I'm going to start by just pointing out that what I'm about to show you is a Jupyter notebook. Here, it's called Vivarium Tutorial. It's been uploaded by Vivarium Tutorial over here. It's not available on Colab, but if you want help setting it up, let me know and I can help set it up with you. Though you should have access to all this code. In particular, I want to point out that there's some hidden cells. You see they're marked for skipping that are just going to help me make this slideshow run smoothly without a bunch of code. So we're kind of, you don't see everything that went into making this code possible just for simplicity. Okay, so the Vivarium coding tutorial. We're going to look at some of the code to run Vivarium processes. This is aimed to be kind of like a how-to guide that's hopefully a reference people can come back to. It's, point out that this does require an intermediate understanding of Python and object-oriented programming, but I've tried to make it as minimal as possible. The outline for this coding section. First, we're going to see how to make a process and add that interface to your model. Next, how to make a composite in which processes are combined. And finally, some more complex composites with multiple different paradigms included. And visit our documentation for more complete instructions. These things are installable. I have some instructions here about how to install and run them. Okay, so to start, how to make a process. These are the basic building blocks. In order to build a process, you take your model and you add the interface with the following protocol. These three requirements. You need a constructor that accepts parameters to configure the model. 2, you need a port schema that declares the ports and their schema. I'll show you what that means. And the next update is the update function that runs the model and returns an update. And so here's our little flowchart symbol for process with all the requirements in there. Okay, going down into make a process, we're going to look at this very, very simple, minimal example of exponential growth, where we have a mass variable that grows as a function of the growth rate and just a little bit of noise. It runs for an increment of time. Okay, so to start, we look at the constructor. First part of the protocol. This is not a working code. It is just a little chunk that shows it. So the constructor, you have default parameters. Whatever process you build, whatever model you have might have different parameters. So you can define whatever parameters you want. For this one, we have growth rate, growth noise. You can pass. So when you make this is the init or the constructor. Which is needed to initialize a process. In order to initialize a process, you just pass parameters in. They go into the super, which overrides the defaults. If you don't pass any in, you just call it with an empty function argument. It would use the defaults, but you can override these by passing in new parameters. Advanced constructor. I'm not going to go into great detail about the advanced cases. Every single one of the protocol can be stepped up a notch and made more advanced in order to personalize it to your needs. Here I'm going to point out that you can import different libraries. So for example, you might import Cell Modeler, in which you initialize it. The required time step, you can declare that and the engine will run it according to the time step that you expect, and you can make your process into a parallel process as easy as this. Okay, next part of the protocol is the port schema. So you require a port schema method inside of your process that declares the ports at the top level and then all the variables that are inside it. Here we just have a single variable called mass, and the schema are these settings that tell the simulation how to handle this variable. You have various different schema settings that you can use in here. Please refer to our documentation for what these all mean and the different options. Advanced port schema, this is all modularity is embraced. You can load in whatever schema you want, not just the presets. So here we're loading a special divider called asymmetric division that is declared right there. This is going to tell the simulation how to divide your molecule. Here we have 2 ports, and we have front and back, and they have one variable each called a molecule. It's going to be divided asymmetrically. Next update. This is the final part of the protocol for making a process. This is where your mathematical model resides. This represents your dynamical model. It takes in the time step, how long to run, and the current states. You first pull out the states at the start. You apply the mechanism. So this is the entirety of the model for this case, and then you return update through the ports. So mass resides under port 1, and then when we have the update, we pass it back in through port 1 to mass, and that's how this runs. Then, of course, at the next time step, you're going to get your new states and perhaps the new time step. This runs repeatedly. Okay, this is the full exponential growth process. This was actually executed, and so this is the one that we're going to be running later on in the notebook. This is it. This is the entire process. So we have our constructor, our ports, and our update. And of course, this can be made much, much more complex. I aim to have this fit onto a single slide, so it's a small process. Okay, and of course, you can run your process on its own. Here we take the process that was just declared there. We pass in some parameters, so it overrides the defaults. And this is a helper function that we take our process, put it into this helper function called simulate process and experiment, give it some settings. These are hidden in a cell above, and we just plot the output. You can see nice exponential growth. Okay, so next up, how to make a composite, how to combine these. So we have our process. Now we want to combine it with other processes. The protocol for making composite, we have 2 functions, two methods called generate processes, generate topology. I'll show you what that means. For this example, we're going to take our exponential growth process that we just saw, and we're going to add some more imported processes. So here we import these processes from different libraries. Vivarium cell gives us derived globals, which calculates shape properties using parameters from E. coli. We have a divide condition process that will trigger division. You give it parameters that tell it when to divide, and then meta division actually does the division. So these are here imported. Composite constructor has its own parameters that get passed in, can override the individual process parameters. Okay, the first part of the protocol is generate processes. All that this requires is you need to return a dictionary of your initialized processes. So here, exponential growth maps to an initialized process. This is the process we made in the cell above. We pass in parameters for exponential growth, and so it's initialized. We pass in parameters for the divide condition. This is initialized. Here we're using the default parameters. For meta division, there's a division config, which is hidden from view that is telling meta division how to divide. Okay, the final part of making composite, you need to wire the things together. So each of the processes that we just declared over here has its own ports. Exponential growth, as we saw, has a port that we declared this, port 1, and we say port 1 connects to a store called boundary. I'll remind you how these look. Okay, so we have a process, we connect it to a store, just like that. Okay, so, and then this other process connects through its port to the same store. So now these things become linked and so forth. Two ports for this one, 2 ports for this one. Some advanced topology. We can use the same method to embed processes in a hierarchy just by specifying a path down. Within organelle, there's a membrane node, and within membrane, there's a lipid. So we're going to connect this process, these lipids, down the hierarchy. You can also go up a hierarchy by using this dot dot. So external is going to go up 1 node into an environment store. And here we have a way to split up the transport port into 2 stores. So glucose external is a variable. It's going to go into an external store called glucose. And this variable called glucose internal is going to go into the internal store, map onto glucose. So we can also use this to rename variables. Whereas process 1 is going to see a variable called glucose external. It's going to plug into a variable called glucose that other processes might see as some other name. Okay, here's the full composite now, which this has run, and we'll be using it in the later slides. So here we have our processes declared, 4 processes. Oh, and just out of view, we have our topology of how these connect together. Okay, we can simulate it. You can see we start off with a single cell or single node with those 4 processes. It grows until it hits that threshold, and then that launches 2 new processes. So now we have 2, this one was terminated. We have 2 processes running in parallel. They grow for some time, hit that threshold, then each of them divide. Now we have 4 processes running in parallel. Or I shouldn't say parallel, but running. They could have been running in parallel if we passed a parallel flag to it. These are running concurrently. So we have 4, and then the divide, we have 8. Okay, we're going to take that composite and put it into an environment. So we're going to plug it into a lattice environment. This is how it's done. Here's grow divide. That's the composite we just made. Here's some details that we're not going to talk about, but happy to address in the Q&A. So we take the lattice compartment, we put our grow divide composite into it, and we run it in time. So inside of this cell is that composite that we just saw. It grows and divides. Now there's 2, and we run it to get all of these running concurrently, many grow divide composites here, all in the lattice environment. Okay, this is the final part of this coding demo, multi-paradigm composites. So in this one, we're going to be taking a CRN model, a chemical reaction network, using Bioscrape, and we're going to take a flux balance analysis process with Cobra. This is constraint-based reconstruction analysis package, popular FBA package. And this is going to be demonstrated incrementally by adding 1 process at a time. So here we create an empty generator, an empty composite, nothing in here yet. We're going to be adding things to it. So first, we're going to add a Vivarium Bioscape process that has the gene expression of LAC-Y protein. And then we have 2 transporter proteins. LAC-Y is a transporter that's going to move lactose across the boundary, but the DNA assembly, the LAC-Y gene, is going to be inhibited by external glucose. You can see over here, we take the Bioscrape process, we load in some parameters. So here we're just giving it an XML file. For those of you who've been to previous lectures, you know how to make this. This notebook actually builds this file, just it's in our view. So we'll load in an XML file. Here we define the Bioscrape processes topology, and we merge it into our composite. So now we don't have an empty composite anymore. We have a composite with a Bioscrape process mapped together. We run it in time and plot the output. And so you can see over here, this is external glucose starts to be eaten up, and internal glucose inside of the cell, inside of the node increases. When glucose is sufficiently low, Y expression is disinhibited. And so we have lac Y expression, which is red. It's probably really small down here. You can't see it. But once you have lac Y, you can start to take up lactose from the environment. Here you see lactose from the environment be depleted as internal lactose goes up. Kind of simple CRN model. Okay, next up, we're going to take an FBA flux balance analysis process. Flux balance analysis is an optimization technique for metabolic models. We've built a wrapper around Cobra. Here, it's just metabolism. This big model database has over 100 of these large-scale metabolic models. We're going to use a metabolic model over here, AIF1260B. It's a very large model, 2,382 reactions for 1,000 genes represented here and 166 metabolites. So we take our metabolism process, we initialize it with this big metabolic model. I should mention it's an E. coli metabolism. And this is just the process. We're gonna simulate it just to show you what it looks like. External concentrations, internal concentrations rise. We get exponential growth when we sum up the masses of all of these internal concentrations. Okay, now we're gonna do that same thing, but add it to that same composite that we saw before, which has the Bioscape process. So we make our metabolism process, we make the topology, we merge it in, and then we run it. And so whereas you saw that the metabolism process on its own grows exponentially, when we combine it together, it grows for some time until glucose nutrients in the environment run out, and then it can't grow anymore because of the way that this FBA, the metabolism model, is connected to the Bioscrape process. Bioscrape actually constrains the FBA solution, so it doesn't grow anymore. And then after lac Y is expressed, growth resumes. All right, and that concludes the coding section.